Hello, good morning. Myself, I am BSR Botany lecturer. Today our topic genetic code. Anyway, to know the complete process of protein synthesis, to understand the complete process of protein synthesis, the basic information about the genetic code is required. Anyway, DNA is a genetic material which carries the genetic information from cell to cell or generation to generation. Yes, DNA is the director. You take this is the DNA strand, 3 prime to 5 prime strand, which act as template during the transcription. So this is the DNA strand. That DNA strand forming mRNA. mRNA always forms in 5 to 3 direction by a process called transcription. On this mRNA, the nucleotides are present always in the form of triplet, that is called codon. Just I am writing some codon, AUG, then triple G, then triple U. Okay. So these are the codons present on mRNA. Especially these codons code for methionine amino acid, glycine amino acid, phenylalanine amino acid. So this is the polypeptide chain. So what I am saying is, in a polypeptide chain, you just look at here, amino acids are present. So the amino acid sequence in polypeptide chain coded by codons present on mRNA. It is mRNA. Okay. So the codons present on mRNA determines by nucleotide sequence present on DNA. What type of nucleotide sequence is present here? A, T, U, A, here C, here triple C, here A. So, amino acid sequence in a polypeptide chain determines by the codons present on mRNA, which determines by the nucleotide sequence present in DNA. So, that is why DNA is the director. Here we should know the properties of genetic code. Properties of genetic code one by one. First one is the code is triplet. The code is triplet. Why? Suppose when you take adenine, guanine, cytosine, thymine, these are the nucleotides. So when one codon contains only one nucleotide, that is called singlet codon. But in a cell, how many types of amino acids are involved in protein synthesis? 20 types of amino acids. So 4 singlet codons doesn't code for 20 types of amino acids. Suppose doublet 4 into 4, how many are possible? 16. So 16 codons doesn't code for 20 types of amino acids. Whereas 4 power 3, 4 into 4 into 4 equal to 64 codons. 64 codons can code for 20 types of amino acids. Remember that one. How these 64 codons code for 20 types of amino acids, which codon code for which amino acid, I explained it already. I prepared one video, you just watch it. But all of you, please subscribe this content, this channel. Then only I will prepare more and more useful videos. Please subscribe all of you. Okay? Anyway, 64 codons code for 20 types of amino acid. Now the statement 64 codons code for 20 types of amino acid. Is it correct truly? No. Why? In this 64 codons, only 61 codons are sensible codons. Remaining 3 are nonsense codons. These 3 nonsense codons doesn't code for any amino acid. What are the 3 nonsense codons? UAA, UAG, and UGA. These are nonsense codons. Another name of UAA, ocre, ocre. UAA, ocre. UAG, amber, amber. UGA, opal, opal. or amber. So UAA, UGA, UAG, all these are nonsense codons which doesn't code for any amino acid. Remaining 61 codons code for 20 types of amino acids. So that is why, for example, AUG code for methionine, GGG code for glycine. So always the codon is triplet. It means in every codon, 3 nucleotides are present. Remember that one. Okay, so then the code is non-ambiguous. The code is non-ambiguous. Non-ambiguous. What is the meaning of non-ambiguous? Here, very important. One particular codon code for one specific amino acid. Methionine always coded by AUG. AUG codon always code for methionine. One particular codon code for one amino acid. It is the non-ambiguous. The code is non-ambiguous. But the code is degenerate. The code is degenerate. Here, important thing, there is a small confusion. One particular codon code for methionine. That code is degenerate means one amino acid coded by more than one codons. So, for example, I am taking serine. Serine coded by UCU, UCA, UCG, UCC. So look at here. Not only this one, AGU, AGC. So all these codons code for serine. It means one serine coded by more than one codon. But always UCU code serine, that is non-ambiguous. Always UCA code for serine, that is non-ambiguous. So one particular codon code for one amino acid. Whereas one amino acid coded by more than one codon, that is the code is degenerate. That is the small confusion in between. The code is non-ambiguous and the code is degenerate. Okay, very important point. And next, the code is universal. The code is universal. It is important. Except in mitochondria and some in protozoans, you leave that one, exceptional cases. Remaining all organisms, you take phenylalanine, always coded by UUU only. In every organism, U code for phenylalanine. In every organism, AUG code for methionine. So that is why the code is universal. Okay. Next, the code is non-overlapping. It's important. The code is non-overlapping. So, for example, I am taking this is the AUG, the first codon, and UUU is the second codon, and the next codon triple G. Okay. So, what they are saying is the nucleotides in the first codon, nucleotides in the second codon are not involved in the formation of new codon. So, for example, here G is there, UUU is there. So, it never binds and never forms a new codon like GUU. The code is non-overlapping. So the nucleotides of the first codon and nucleotides of the second codon never involves in the formation of new codon. That is why the code is non-overlapping. Remember that. And always the starting codon is the AUG. The first initiator. AUG is the initiator. AUG is code for methionine. Remember that one. And terminator codons. What are the terminator codons which terminates the protein synthesis? UAA and UAG and later on UGA. These are nonsense code. So these are the properties of GENRTIC code. Thank you to all. Please subscribe.